So following glycolysis, we have per sugar molecule 2 molecules of a 3-carbon sugar called pyruvate. And if oxygen is available, we then proceed to the link reaction. And the link reaction is summarized here, courtesy of BioNinja. And what we can see is that the pyruvate is formed in the cytosol or in the cytoplasm of the cell. Following glycolysis, it's a 3-carbon compound. And what we're going to do initially is we're going to take away a carbon. We're going to decarboxylate the pyruvate, which then forms a 2-carbon compound. So in so doing, we release carbon dioxide. So this first process is decarboxylation. Then what we do with the 2-carbon compound is we take away hydrogen. So we are oxidizing that sugar. The 2 hydrogens which are removed from the sugar are given to NAD. So NAD is reduced into NADH plus H plus. The 2-carbon sugar is very unstable, so it needs to be bonded temporarily to coenzyme A, and the 2-carbon compound that is produced is called acetal. So we're going to produce, because we've got 2 pyruvates per glucose, we're going to produce 2 molecules of acetal coenzyme A. We're going to release two molecules of carbon dioxide, which ultimately we breathe out, and we're going to produce 2 molecules of reduced NAD. This is also shown visually here. So oxidative decarboxylation, again, that's part and parcel of the link reaction. The NAD, which is reduced, is going to go on to the electron transport chain, which we come back to in a later video. And the yellow circles here are modeling carbon. So you can see that it's a 2-carbon compound, which is held onto by this cofactor called coenzyme A. Here we've got a summary of what's going on, and this is very much similar to what we've done in the first unit. To add to that, we need to appreciate that what you can do, the details are not required for your course, but what we can do is we can chop up fatty acids, which also are going to allow us to produce acetyl-CoA. So fatty acids can be used and fed into the link reaction and the Krebs cycle, etc., etc. So we can metabolize fat and we can use fat to produce ATP as well as sugar. So once we've done the link reaction, what we then do is we move on to what's called Krebs cycle, a metabolic hub. And here we can see this happening as a summary. So what we're doing is we're going to take the acetal, the 2-carbon sugar, and feed it in. And also what we've got is a 4-carbon sugar, which is at this stage here. So we've got a 4-carbon sugar plus a 2-carbon sugar coming together to form this 6-carbon sugar, which is then decarboxylated. So decarboxylation, we're going to remove carbon dioxide. That gives us a 5-carbon sugar. The 5-carbon sugar is oxidized. We're going to remove hydrogen and we're going to give it to NAD. That 5-carbon sugar is further oxidized. We're going to produce more reduced NAD. Then we do decarboxylation again. Using substrate level phosphorylation, moving phosphate back and forth to a group here, it allows us to have the energy that is going to produce ATP. And then what we're going to do is, because we decarboxylated here, have a 4-carbon sugar. The 4-carbon sugar is oxidized. This time we're going to produce reduced FAD. Then we've got further oxidation. So we've got oxidation happening at each of these steps. The sugar is oxidized and we are either going to reduce NAD or FAD, and we're going to do decarboxylation. So again, we need to know the names of the reactions that are taking place. And ATP, it's not going to make sense to you because there are parts here, details which are missing, but we're doing phosphate substrate level phosphorylation. We're taking a phosphate group where it can give it to the sugar here and then pass it back to ADP down an energy gradient, and that just allows us to produce this ATP. And remember that per glucose molecule, we've got 2 pyruvates, which means we've got 2 acetyl coenzymes, 2 coenzyme A, which means that Krebs cycle turns twice per glucose molecule. And again, you need to make sure that you appreciate that all of these reactions are enzyme controlled, and the enzyme is lowering the activation energy. The enzymes are also able to, they've got an active site, so link everything to 8.1 essentially. And then here we've got further links and connections, and have a look at these which are posted in the Google Classroom in the PowerPoint and explore that to your heart's content. And then here again, we've got a summary from these circles. These yellow circles are meant to represent carbons. You can see the decarboxylation stage. Remember that all of these reduced NADs are moving into the electron transport chain, and Krebs cycle is taking place inside the mitochondrial matrix. So the hydrogen ions and electrons, so hydrogen is removed from the sugar, which is going to be split up into hydrogen ions and electrons in the next part. Here we've got a summary of Krebs cycle, so pause here, read through and make sure that you're clear. And in the next video, we're going to look at the next stage, which is the electron transport chain and chemiosmosis. This is a bit more complicated.